Hello students, so now we are going to discuss about the cultivation of medicinal plants, right? So before going into the topic, first we need to define that what is the cultivation. So cultivation is a process of preparation and use of land. What for? For the growing of plants or crops, right? So what we are doing here, a process of preparation, whatever we maybe plugging up the soil, maybe by using of the different tools and machineries, we are preparing the soil and using the land for the growing of the plant or medicinal crop. Okay, now so what are the advantages and disadvantages? That's very important. Why we are going for cultivation? What are the advantages behind that? So the first advantage is their quality and purity. We are going to ensure the quality and purity of medicinal plant can be assured by the process of their cultivation, right? Since because we are going to take care about the water supply, irrigation, fertilizers, pesticides, and interculture property, distance between one plant to another plant, all those things we are ensuring, and we at the same time we will ensure the quality and purity. Second advantage we can mention here, drug with the highest content of active constituents, that is possible only with the cultivation. For example, gingiber officinalis, gingiberol we are expecting from here, that can be possible. That gingiberol is the active constituents in this what you call volatile oil is active constituents, turmeric, curcuma longa, right? Curcumin is the active constituents in turmeric, that can be possible only with that. Glycyrrhiza glabra, licorice root, that is glycyrrhizine or glycyrrhizic acid is the active constituent, that is possible only with the cultivation. It can be obtained by systemic cultivation, right? The third advantage we can mention here is production of drug of good quality is possible by the maintaining uniformity in all operation, like maybe application of fertilizers, water supply, irrigation, interculture property, that whatever the hectare air area of the land we are using, there we are going to maintain the uniformity. Fourth advantage you can mention, because of well planned cultivations, regular supply of crude drugs to the industries is possible. Ultimately, when it will be in the large scale production, large scale cultivation, we are not going to use it for the our household, right? Ultimately we are supplying is the industry, and industry supply, it is possible something, suppose any industry required 1 ton ginger in per day, so that is possible only with the cultivation. We cannot depend on the wild source or cultivated source. Fifth one you can write down, rapid growth of phytopharmaceuticals, rapid growth of perfumery in industries is possible due to cultivation of medicinal plants, right? So that you can mention these 5 advantages, but at the same time we are having some disadvantages also. So what are the disadvantages? When we talk about disadvantages, the cultivation of medicinal plant is costly compared to the wild grown plant. Of course it is costly. Second disadvantage you can mention, costly due to the loss of drug by natural hazard. That's very unexpected thing which happen generally. It may be storm, it may be floods, hurricanes, earthquake, landslides, etc., which is unexpected. Natural, by nature it will be your plant is going to damage because of heavy rain, flood and all those things, right? So nowadays anyway crop insurance it is available, and government is reimbursing the amount whatever you have invested. If you have done the insurance for your crop, that's different thing, but these are the disadvantage you can write down. Okay, now coming to the cultivation techniques, when we talk about what are the techniques it is there for the cultivation of medicinal plants. So when we talk about the cultivation techniques, we are having certain things here to discuss. For example, 2 types of cultivations it is possible, right? The first one, 2 kinds of cultivation techniques, seed propagation and vegetative propagation. Seed propagation is called asexual method, and vegetative propagation it is called sexual method, right? So first one we will discuss seed propagation. So in this technique, new plants known as seedling are grown by the sowing seeds, right? So new plant it will be grown by the sowing seeds we are going to use to grow the new plant. So there are 3 methods. First one is the boat casting method, right? So in boat casting method, in this method is used for the seeds which are extremely small in size, right? What is the condition here? Extremely small in size seed, like mustard or something like that. Okay, this is done by the help of seed drill. Seed drill we are going to use, so that uniform distribution or replacement of the seeds it is possible with that. the seeds are spread over the surface of plugged soil, right? Whatever soil it is plugged, in that surface we are going to put the seeds uniformly, right? So since these are very small in size, something we have to take care, right? So what are those things we are supposed to take care, right? If the seeds are sown deeply or covered by thickly by soil, what will happen? Then they don't germinate. Why? Due to the lack of seed respiration, right? Seeds are going to have the respiration, they need oxygen, the passes of. So if you will cover deeply and because they are small in size, if they are very inside, and thickly if you covered by soil, definitely the seed respiration will not be there, and they will not germinate. Like in case of that's what spacing is very important, preparation and uniform depth is very proper, and uniform depth is very important, and proper moisture zone the seeds has to be placed, right? So what are the things we are doing with this method? Rice, wheat, methi, coriander, sesame, these are the example for the this thing. Second method is dibbling method. Dibble the seeds in the holes, right? Dibbling. So average in size of the size and weight of the seeds can be used by this method. New plants are grown by the sowing seeds, yes. Shown by placing in hole of ground, like there will be the hole, like different holes will be there, in that we are going to show the seeds, okay. In each hole 2 to 6 seeds put together, like carica papaya, you might have experienced also, right? Papaya seeds we can put into the hole, castor seeds and fennel, these are the seeds which are average in size, that can be dibbled into the different holes which has been made, right? Seed can be dibbled at the desired depth in moisture zone, right? That's what optimum plant population can be obtained if you are having the proper distance hole. Other method, third one, other method you can have, or it is called a miscellaneous method, right? That also can be mentioned here, okay. So when we talk about the other method of this thing, we are having most of the seeds are grown by sowing seed in nursery beds, right? Nursery beds we are going to first we will grow, and then the plant are then transplanted to the farm or open field for further development of the plant. For example, cinchona directly. In rice also we are doing in the same way, right? First we will put into the nursery beds, then we will transplant into the open field, like cinchona digitalis, it is there, cardamom, clove, etc. That's what CCCD, triple CD you can say, cinchona, cardamom, clove, digitalis. So these are will be done in the nursery bed method or miscellaneous method. Few things we need to have here take care about seed when we are going for seed propagation or asexual method of cultivation. The stimulus for seeds germination, what we are supposed to do, the pre-treatment, some special treatment given to the slow germinating seeds for the to increase the rate of germination. For example, complete immersion of seeds in water for a day or maybe 24 hours. We have to deep into the water, right? The seeds like castor seeds, we cannot directly go for the showing them, right? We have to treat, this treatment is important. Second thing, immersion of seeds in H2SO4, in hand bend seeds we have to do, right? That's what immersion is very important. If in this kind of acid, if you are immersing there, the transparency, the seed coat will become soft, and the germination will become easy for the plant. That's very important, we need to take care, okay. Then further we can have treatment. That treatment it is like for example, to improve the germination rate, removal of testa, it's very important, of seed, because testa is the outermost layer, okay, outermost layer of the seed, which is impermeable of water and gases. So in Indian senna we can remove the testa, so that the seed respiration will increase and germination rate will get in good this thing, okay. Use plant growth regulators, PGR, right? Sodium hypochlorite can be used, ethylene, cytokinin, potassium nitrite, thiourea, oxine, gibberellin, this can be used to increase the germination rate. And most of the thing, this very important, we need to take care, long storage seeds to be avoided. If you store the seeds for long time, maybe 2 year 3 year, the germination it will not be taken place, because the number of viable cell it is going to decrease, right? So 1 or 2 year it's fine, it depend on the crop to crop. Now second method of cultivation, it is vegetative propagation, right? So when we talk about the vegetative propagation, it is also known as asexual method of cultivation. New plants are raised from the vegetative part, maybe stem, maybe root, maybe wood, buds, flower, something like that. First one is the natural method and second one is the artificial method. So under natural methods, we are having example, for example, garlic and onion. So garlic and onion, it can be cultivated by using of bulbs. Okay, saffron we are using corms part, turmeric we are using rhizomes, rose we are using stolons, pineapple we are using suckers, and in potato we are using tubers. Coming to the artificial method, we are having so many things like grafting, budding, and all those, right? So the first thing in under artificial, cutting. Okay, so the portion of the plant which capable for developing into new plants are separated. Any portion, it may be root, stem, bark, right, from the plant. The portion of the plant capable of developing new plant are separated from the plant by cutting. That's what name itself is, cutting. By cutting, the separated portion is placed into the soil, right? Just in rows we are doing, right? That's what we are when we are going somewhere for just sightseeing or something like that in anyone's home. We, when we get some good quality, good color of the rose, we are taking one small branches, right, for the root development, then we are placing into our home for root development. So this thing will be done, the cutting will be done in the rose, orange, even in citrus plants like gulab, santara, nibu, something I have written in the, so that, okay. Then second thing we can do here is layers, right? Second method we can do is here is the layers. So when we talk about the layers, this is also one of the technique. Parts of the plant that has grown out from the stem, like branch, that has to be partially slit, right? For example, this is the branch, this is stem, so this has to be partially slit. It will be there in this way, right? So partially it has to slit. So the slitting has to be done in a such a way, slit should be made such a way that expose the inner tissue should be there in that branch, okay, or any piece of wood part also, so that, and then it has to be transplant into the open field, add plant growth regulator, develop root, then it will go for the root development. We are doing this layers technique in the lemon and jasmine plant. So example is very important, that you have to write compulsory. Many students, they will write about layer, grafting, all those things, but they don't write the example. That's very bad thing, and you have to write, you should write, then you can get the good marks. Okay, then second thing, it is the grafting. So when we talk about the grafting, what we do is piece is cut and removed from a living plant, fixed in a cut. So in grafting, a piece is cut and removed from the living plant, fixed in a cut into the another plant, right? So there will be two plants, that's what it is called a column, one plant and another plant, right? So from here one plant pieces will be cut, any branches, and it will be fixed in the another plant. That's what we do, fixed into a cut into the another plant, so that two cut portion or surface grow together. So here the in new plant where it will grow, and whatever the fruits and everything we are going to get in this plant, that will be having characteristic of the parent plant, like cinchona species. So here you are in grafting, you are going to give the example of medicinal plant, right, not the other which you do regularly, right? Cinchona species you can do. Fourth one, budding. T-shaped cut is made, right? In this case we have to make the T-shaped cut into the plant. A cavity is made in the stalk bark, introduce a piece of bark, consist bud. Okay, that's what our intention is, budding. So that's what budding will be done, right? Budding will be done. Citrus species, sweet orange, and all those things can be done. Fermentation, chemical breakdown of the substances by bacteria or yeast and used in manufacturing of antibiotics. That is also one of the cultivation techniques. Then we are having inoculation. It is the introduction of microorganism into the living plants. For example, in ergot plant, fungus spores are injected into the plant, so that is also another technique, right? Apart from this, we can go for the another techniques that is called in this only cultivation, that is a micropropagation, right? So when we talk about the micropropagation here, micropropagation of aseptic method, right? So like this. So micropropagation, as the name indicate, right? Micropropagation is what you call here, you can see micropropagation is new method of production of medicinal crop. Fine segment we are taking, single cell, maybe callus, embryos, root tips, shoot tip, ovaries, any parts, right, of the plant can be taken, is developed into a new plant where in aseptic condition, under the aseptic condition in artificial medium, right? Just like in microbiology we have done the cultivation of bacteria, like that nutrient media will be there, and the different parts of whichever is having totipotency capacity having to generate the new plant that we are taking, and in aseptic condition means free from microorganism condition in laboratory environment we are going to grow, right? That's called here we need to have the nutrient medium, like that we are going to take in the detail in the plant tissue culture unit 3, but even though here also you need to write nutrient medium preparation, we need to have the inorganic solvent, microelement, macroelement, carbon, oxygen, calcium, magnesium, iron, potassium, all those things, carbon source, activated charcoal, vitamins, plant growth regulators, oxygen, gibberellin, cytokinin, and all those things we need, right? Organic supplements and all those things can be added. Medium sterilization is very important, necessary to sterilize the medium to decrease the growth of microorganism. Autoclaving we are doing at 15 lb pressure at 15 to 30 minutes time in 121 degree centigrade temperature, that you know very well. Pressure cooker can be used and hot air one can be used for to sterilize the glasswares, right? Explant preparation, what we can do, explant preparation is isolated part of the plant is placed into the artificial medium, right? Any root, stem, fruit, flower, bark, it has to be placed into the artificial medium, and surface sterilization can be done for the plant part used, right? We can wipe out by using of different chemical like sodium chloride 0.1 to 1%, right? Silver nitrate, then sodium hypochlorite, sodium peroxide, it is treated with 70% of ethanol for 2 minutes also can be done to remove the microorganisms before going for the plant tissue culture, right? So that is also important here. Explant inoculation, what we can do here is explant inoculation, introduction of sterilized plant part into the nutrient medium under aseptic condition, okay? Sterilized plant part which we have done just now with the surface sterilization, no, that we have to put into the nutrient medium under aseptic condition. This process is carried out in laminar airflow chamber, not in any bench or somewhere. Laminar airflow is already aseptic area, or sterilized inoculation chamber, it depends where whatever the facility available with you, that can be done, right? Now inoculation, it is very important to understand. First of all, in incubation, first we need to take the container with the nutrient medium and explant, though both has to be mixed in the proper container, then incubated in incubation chamber, like incubators we are having in the bacteriological incubator is there, no, like that incubator where we can maintain the temperature 25 degree centigrade plus or minus 2 degree centigrade temperature, humidity above 50%, and then after 3 to 4 weeks the callus grows 5 times than its actual explant. That growth will occur because of the nutrient they are going to use and they will develop, right? So once they will develop, the grow has been done, what we can do is we can add the plant growth regulators, and the complete plant can be transplanted into the open field or somewhere wherever you want to grow, right? So these are the 3 types of propagation we can have First one is the seed propagation, second we have discussed the vegetative propagation, and third is the micropropagation. These three is the method of cultivation, right? Thank you.